Good afternoon. My name is Marcela Karey Tello-Ruiz. I am the project manager and outreach coordinator for the Gramene database. And as you know, Gramene is a plant comparative genomics database. But today we host over 50 genomes, but today I will focus on resources that might be of special interest to maize researchers, but can be applied to many other species. I will start by telling you, if you're not aware already, that the project is a transnational collaboration, and this is the people that make it possible. I am personally based at Cold Spring Harbor Laboratory in New York, but we collaborate very closely with the JSWA Lab at OSU, with the Ensembl Plants and Expression Atlas at the European Bioinformatics Institute, and the American Society of Plant Biologists. I will begin by telling you giving you a little overview and where to find additional educational materials for your own exploration. I will show you a short video of the Gramene search, which is the main entry point for the website. I will spend a little more time on describing the Genome Browser and give you a little flavor of what our latest, one of our latest ventures, the Midspan Genome, is. And I'm going to leave you with a short video by Dr. Synteny on the pathway portal of Gramene, the plant reactor. To go to Gramene, you simply type www.gramene.org, and you will see the homepage with little icons for the different aspects of the website that you can find. On the right side, you have our news blog, and in the link for today's webinar, I will post the slides for these and some exercises that you can practice with for today's webinar. In addition, you saw over here, we have an outreach and training icon. This page includes some additional links where you can find more information about the website. And in particular, we have a link to our YouTube channel where this recording will be posted a posteriori, and our FTP where I will also place the slides. In brief, here is a little overview of what you can expect to find in the database. We have currently 53 genomes in the database, over 2 million gene annotations, 67,000 gene trees, and at the DNA level, pairwise whole genome alignments across the species. We have expression data in conjunction with the Expression Atlas at GBI. We have pathways data curated in RICE and projected based on homology to 74 additional species, including the 53 reference genomes that we host. And we have variation data in which we have brought into Gramene through the European Variation Archive. All of this is integrated through our custom search and visualization tools, which is the short video that I'm about to show you, and I may want to pause it a little bit to tell you a little more detail. So from the Gramene website, you can press over here this little help. This little question mark will show you the different features that you can expect in the search. As you type, you're going to be given suggestions for what you can expect to find, and the results are going to be ordered by category. You will see the results, the hits, organized also by species, And each one of these results is going to be centered around a gene view. So as we proceed, I'm going to use the example of a pathway, the carotenoid biosynthesis pathway. I find it here under the plant reactant pathway category, and I see that there are hits in all 53 genomes. There are 428 genes. I am going to, since I don't want to go over all of those genes, but I'm interested in the maize genes, I'm going to select C maize. I'm going to type done, and I see that there are only 7 genes in the maize species. Among those, I am going to focus on this one, yellow endosperm 1. And before we go any further, you can see that there are 5 different tabs. And the video is going to go through each one of them. The first one, location, will give you the genomic context of your gene of interest. Here are visualized exons and introns. And you will also see links to other resources. And the top one is the Ensembl Browser, which is the one that is the internal one that is the one we use. And you will see a little more detail later. But this is just a snapshot of the different things that you can find in the results. Right away, you can see whether you have genomic context, expression data, homology, gene trees, pathways, and cross-references. So, oops, I'm sorry. Let's go back here. So we were about here. Okay. Here. I think, yes. We have selected the maize gene. We go to the yellow endosperm. We see location. We open up location tab. We see the different tabs. We see the phytomine and maize GDB, other resources for maize. Under expression, we see 2 views. This is the whole organism, and we're going to also see the reproductive parts of the plant. And you can see over here on the left is the study that reported this data. You have the various tissues over here on the top. And you have other variants, like they are color-coded by intensity. You can filter sort by experiment. We can for other filters, and you can download all of this information. When you mouse over any one of these parts, plant parts, you will. That's what you. It highlights the actual study. Under homology, you have, in this case, I'm only seeing maize. But if I go back and ask to be all the species to be shown and focus on that one gene, the yellow endosperm, you'll see that one over here. I'm going to see all of the species. Now I have a single hit in maize for that maize ID, gene ID. And under homology, now I'm going to see all 53 genomes in Gramene. Okay, this is a little tricky whenever you do recording, so I'm just gonna jump in here. That's the whole tree. Why? It's so sticky. Okay, so you see the whole tree, and you have different views. The first one was the whole view. This is multiple sequence alignment, which is a zoom-in view at the amino acid level. You can drag your slider and see reposition to see the conservation at the amino acid level across all these 53 species. You can also change the color, the scheme of the colors. And another useful, you can also add the species. You saw before we only have IDs. Sometimes IDs alone are not as informative, so you may want to add the species name. And another type of view is the neighborhood conservation. I'm going to pause it here for a second to show you how you have maize, and the closest in the tree are sorghum and setaria, but they are not being as informative for this particular gene. However, if you look down at one of the rice homologs for this gene, you'll see that in the several Oryza species, all these 11 species or so. You see conservation of these. Each one of these represents one gene to the left or to the right, flanking genes around your gene of interest. So let's finish this. Under pathways, remember we started from a pathway, the carotenoid biosynthesis. So now this view, what this view does is show you exactly in which reaction of that pathway is your gene of interest actually acting. And any of these snippets is linked to the actual resource. So from here, you can go directly to the plant reactant website and find out a lot more information. Under cross-references, you have additional UniProt, UniParc, ENA, in addition to those that you saw in the cross-reference view. Oops, I'm sorry. Okay. So here is a summary slide with all the 53 genomes currently hosted in Gramene. These 8 ones over here were added within the last year, and these genomes, these assemblies, or the gene annotation for those assemblies were updated as well within the last year. So remember, this was our home page, and if what you just most of the images that you saw come from the Genome Browser. You can access it directly by clicking under Genome Browser, and we will be taken to this genomes page, which is something that we produce in collaboration with Ensembl plants, and we use the Ensembl software to project this data. So you have, in this release 57, there is a section of what's new. You will find that in this release, we have new polyploid views, which apply very well for wheat, in the future for other genomes as well. We have new rice genomes, rice gene synonyms. We have SIF scores for a lot of these species displayed, and as well downloadable. And but if you want to see all of the species, another form to see all of the species in addition to maize, you can click here to go directly to maize, or you can view the 53 species when you click on that link, and you can find also maize here. And what this table shows is a summary also of the different kinds of data that we have for a particular genome. For maize, you see most of those are marked. So we have variation, we have regulation, expression, we have whole genome alignments, and peptide compara, homolog, orthologs, etc. So when you click on any of those See Maize icons, you're taken to the maize species page, and this has other various sections. You can find out more about the actual assembly, the current before assembly in this section, or if you want more about the actual gene models in this assembly, you can convert your old assembly IDs, the Grimson IDs, into the ZM 00 something new IDs. With this table, you can also take a quick look at the gene function summary table. You can download the transposon data from this link as well. And as you see, any of these sections had little icons with an example of what a particular type of annotation looks like in the actual Genome Browser. So now let's go to an actual view of the browser. And over here we have additional links that may be interesting for you. We are going to navigate the Ensembl Browser, and this is the from the maize species page. You'll see that when you look for a gene, you're going to be given a location tab, which has contextual information for your gene of interest or for any given region in the genome. You can have also the actual gene tab, a transcript tag among for any particular transcript for that gene, and you can have a variant tab for a single nucleotide polymorphism as well as a structural variant. So each one of these tabs will have a different set of options on the left menu, and as you travel, you'll see, for example, the first one, you can view Synteny, so the entire chromosome, at the chromosome level, you can see alignments. You can narrow and go to alignments at that particular region. You can see comparative views, it's, entire regions compared. When you go to the gene view, then you have other options. In what I'm showing here is an image of all of the SNPs in this particular genome, the yellow endosperm that I use as an example. They are color-coded according to what functional predicted effect they will have in the protein. And in the context of interprodomains, as predicted by various software programs. But you can also see from here gene trees, genomic alignments, and other interesting information. At the transcript level, again, you can also see for a particular variant protein information, SNP variation in the form of a table or an image, and various. So it's up to you to explore. For a variant, you have other options like in this case, I'm showing the flanking sequence. Here is the focus variant of interest is a serine S, and all of the variants in the sequence of this gene are also color coded by functional effect. But you can also see in some cases genotype frequency. For this case, you can have some sample frequency in other for some of the data sets. We do have certain limitations for numbers. So for Maize sets, it's difficult to see. But if you go to sorghum, you will be able to download actual individual genotypes for individual samples. To configure browser, any of these images, you'll see there are some symbols that are common to all of them. In this case, you see that spider gear. It's a common symbol to indicate that you can configure that image. You can, for example, turn it off, turn the entire track off. In this case, I'm taking one of these. These are, if you click on a particular track, this is what you will see. So you can configure, you can show different, it will display in different ways. So you just have to try the one that suits your purposes better. For variation, we're teaching. We currently host three kinds. You can say three kinds of variants. We have the single nucleotide polymorphism naturally occurring. We also have induced by mutagenesis, EMS, but for sorghum and wheat, and we have structural variants currently only for Arabidopsis and sorghum. You may think, but this is not relevant for maize, but we actually think that this information in sorghum can be very useful for maize researchers, and I'm going to give you a couple of examples. This is just a summary of the amount of data that we have for these 9 species, about 230 million SNPs. For maize, we have 3 main sets. Well, actually, yeah, two and one more coming. And I'm going to the next small snippet of information is about the EMS-induced mutants in sorghum, because we consider sorghum as a functional genomics platform for the maize community. This mutant collection is described in this paper by our colleague Yinping Yao, and and you actually can order seeds from that you're interested. But how do you go about exploring this collection? Let's say that you are interested in finding any EMS mutant that produced resulted in a truncated version of your protein product. So you're going to look for stop-gain alleles in, say, transcription factors. So you're going to go to Gramene and you're going to select different filters, attributes. Here is the example, and you will get a final data set of such transcription factors in the EMS collection. For those, you can explore them further in the genome browser. The various views, you can see different information. You can see expression. You can see methylation patterns. You can see them family trees. You can see the variants in the context of protein domains. Or you can actually focus on a specific variant, gather more information. In this case, I'm showing the genotypes for particular samples, and you can order those. Another example, it's from the same group, sorghum, in which we collaborated. Sorghum, this is a mutant that tripled the number of seeds in the plant. So you may be curious to see whether this is something conserved in maize, and you want to explore this gene in our tool. So the first thing that occurred to me when I look at the paper, like, oh, unfortunately, these are old. They used older coordinates in an older assembly. So the first thing I need to do is to convert them to the current assembly. So to transverse from one assembly to another, you can use the assembly converter here. If you have a sequence, you can also use the some of the BLAST or the BLAST tools. You can use MMR to also BLAST protein sequences and other things. So here is for that particular example. Remember, this was this will be table 2 from that paper. This is the variant image that I gathered for sorghum, for the sorghum gene. And this is a variant table for that particular. I see that there are these mutants in the EMS collection. Unfortunately, the mutants reported in the paper are not in the sequence population. So we cannot see them here. But the question would be, is the amino acid sequence conserved in other species like maize? And that's how you can go about using sorghum as a platform for your own studies. You can, for example, look at the Synteny for Synteny maps for between sorghum and maize for this particular region. You can also, under that location view, line up sorghum versus maize and see whether there are conserved SNPs in here. You can color code them and you can link. So it's a very useful resource for comparative, to do comparative analysis. You see here is the sorghum gene, and this is the homolog at the region, at the corresponding regions. Track hubs, this is another another feature that in Gramene that you may be interested in, and it is the registry. How you go about it, you're going to go to the location view and click on the configure this page. Under personal data, you're going to search for public track hubs for maize. And when you do that, you get a selection, and you can choose whatever study that you're familiar with. You can project. You can display in the browser, and here's an example. In that, it's following a very similar process. You can upload your own data and compare it in the same image and make your own figures. Now I want to switch quickly in the last few minutes. I want to bring the project, the Midspan Genome. This is independent from Gramene, but we at Gramene we have a pan genome for preview at maize-pangenome.gramene.org, and the project is about sequencing, assembling, analyzing the 24 maize founders. You know, currently the maize reference is B73, but a lot of research has gone into these 24 founder lines, and the goal is to have them all in one single genome browser by next year. The chromosome, the entire assembly with their gene annotations, with comparative analysis like the gene trees, the Synteny maps, the pan-gene, and our tools will allow to answer questions as simple as copy number variation, presence, absence. Remember this gene neighbor view? You can see whether in the different, compared to sorghum or the reference maize, whether those a number of genes surrounding your gene of interest are conserved or not. Or that is at that would be at the protein level, but you can also see at the DNA level whether the actual sequence is conserved or not. Currently, we have only 3 lines on site on this sub-site, we call it, but we have placed it in a very relevant species for you to have an idea of how they compare, how they look in the evolutionary ladder. And be able to make your own protein and DNA genome comparisons. So similar to Gramene, the Midspan Genome allows you to have gene tree alignments. Here, these can be chosen under the homology tab of a search. You can choose between 3 different kinds of displays. Here is shown the gene tree view, but you can also go to the multiple sequence alignment here under the display mode and look at the amino acid level to see conservation, whether any region is missing or absent on a particular genome. And again, the gene neighborhood conservation view will allow you to see whether entire regions or genes whether are missing or which could be either an annotation error or could be an actual biologically relevant difference. And that would be all for genomes. But now I want to show you a short video by Dr. Synteny. I hope the volume is fine. If not, I'm going to get out of this and project the video directly from. No, I think I have to get out, and I'm going to open the actual video. Let's see. No sound, huh? Why? Okay, well, I can try to describe what she's showing you here. The plant reactant you can access directly from Gramene under the pathways icon, or go to plantreactant.gramene.org. These are, as I mentioned before, we have curated pathways in rice, currently over 270 or so. Orthology-based projections for 74 additional species. Here she's showing for Oryza. We have the pathways are organized hierarchically. You have reactions at the granular level of a reaction. You will see over here all the molecules. You can download all of the information that you see. Here, this is a fireworks pathway hierarchy display, how they all connect pathways. This is for a is showing here. It's a particular reaction in the inflorescence development. Just the way I showed you before, when you in the search view, a particular reaction where it happens, it shows in the image as well as in the network over here the reaction. Again, here is just another picture. We are integrating data that comes from Ensembl, from Atlas, from collaborators. Here is the molecules, 21 molecules in this particular, participating in this particular part of the pathway. There is, you can share this, and you have this. The newest thing is the plant interactome. Over here, this is an example with Arabidopsis. There is the source. I am not sure if this is ready for maize, actually, but that's something that maybe Dr. Neitani, if she's in the audience. Yes, Dr. Neitani is in the audience, so if you have any further questions, she will be able to help us with that. I think, well, this is more for the analysis. Under analysis tools, you can have, you can overlay your own expression, custom expression data, and compare it among different, with other species. The pathways, here is a scale for the intensity of expression. In the same way, you can see that in the table. The reactome has APIs and the widgets that we share with other with our collaborators. That was the expression data, and here is a little commercial for the upcoming Pathway Jamboree in the context of the what is it? ICBO. I can't remember the exact, the International Conference on Biological Ontology. For curators, you are welcome to join. We always, we always appreciate anyone willing to help us with our curation's efforts. So I am going to, I'm sorry, just finish by telling you how you can get in touch with us. I will post the slides in this link and also under the webinars in the FTP. We have the outreach where you can download other educational materials. You can email us. You can contact us through the feedback form. You can go to our YouTube channel or read through our news blog, or of course like us on Facebook or and follow us on Twitter. And I'll leave you with the people that makes this possible. And I am open. We're a little over time, but I'm happy to answer any questions you may have for me or for Dr. Neitani. I don't see any questions. Oh, Sushma. Thank you. All right. Well, we're here. And the slides and the exercises will be available on the FTP this afternoon. The video recording may take a little longer, but it will also get published on our YouTube website channel. Thank you.